Good afternoon. Thank you for organizing this talk. My name is Marnye Isaac, and I'm a professor and Canada Research Chair at the University of Toronto. My research group draws on an interdisciplinary approach and makes use of empirical evidence from agroforestry systems to describe the drivers and diagnostics of diversified agroecosystems and the role of biodiversity in governing the structure and function of these systems. We predominantly study coffee and cocoa agroforestry, these diversified systems that are now more than ever recognized for their transformative potential in sustainable agroecosystems. These farming systems are experiencing a watershed moment, framing a critical lens to evaluate agricultural sustainability. Underpinning these transformative pathways to sustainable agriculture is the diversification in agroforestry systems in order to replace external inputs with biological and chemical processes to create beneficial trophic interactions and to add to crop portfolios. We know that diversified agroecosystems such as this have many ecological benefits without compromising yield across scales of integration, including a suite of ecosystem services seen here on the screen. While we've gotten very good at measuring services provided and responses in these systems, we still largely and quite narrowly use approaches to describe plant responses to environmental conditions in agroforestry systems. We really use three dominant paradigms to do this. Agronomy, which evaluates crop response to experimental manipulations of the local biophysical environment. Process-based modeling, which assesses crop response to environmental stimuli based on physiological rates of a plant, as well as landscape ecology, which really couples larger scale land use patterns to the environmental and socioeconomic outcomes of agricultural practices. Now, while these approaches, of course, have contributed substantially to our understanding of agricultural systems, we really want a new approach to assess mechanisms such as optimal acquisition strategies and breeding effects on crop biology. And to do this, we're using a new framing, plant functional traits, to generate new hypotheses on the consequences of breeding on crop response in agroforestry systems. Now, plant functional traits, distinct from agronomic traits, really focuses on the cumulative hypothesized influence on the structure and function of an agroecosystem. And functional traits or suites of traits can inform our mechanistic understanding of plant response to management and environmental stimuli. Now, what makes functional traits so useful in agroecosystem management context is that nearly all groups of functional traits have been hypothesized to co-vary along a distinct spectrum, a universal axis of resource acquiring traits to resource conserving traits, thus describing the functional variation among plant species and within a species. And this concept of trait spectra was really popularized by the notable leaf economic spectrum. Within the leaf economic spectrum, we have species or individuals that fall on an acquiring end of the spectrum, expressing leaf traits such as high photosynthesis, high leaf nitrogen, and high leaf area. And on the other end of the spectrum, we find leaves or species that are expressing low photosynthesis, low leaf nitrogen, and low specific leaf area, all representative of conservative traits. And this variation can happen within a species or across species. This body of literature on functional traits, coupled with commodity-based studies, have really indicated that this trait-based approach research can make significant contributions to understanding and predicting the effects of selection, crop expression, and resource acquisition in agroecosystems. Now, I'm going to draw on our studies over the last 6 years to illustrate where and how leaf and root functional traits can effectively diagnose and predict crop response. Our first studies in this area really focused on coffee leaf traits, and that's what I'm going to focus on for the next few slides. In this study, we were very interested in the overall trait variation of coffee leaves in agroforestry systems along a gradient of climatic and edaphic conditions. This figure in front of you is showing photosynthesis on the y-axis and leaf mass per area on the, photosynthesis on the x-axis and leaf mass per area on the y-axis. We can see the distribution of individual species along this axis, represented by the black dots, about 700 different species, and the red dot representing the mean value of coffee leaf expression of photosynthesis and leaf mass per area. Now, this mean dot is often taken as the extent of trait expression of coffee, but when we actually plot all of the values of trait expression of coffee in these agroforestry systems, we can see high variability in trait expression, following similar patterns we'd expect from among species, but with high variation. What this has led to, after researching this across other species, is that we find that breeding has really led important crops to express some of the most extreme leaf functional trait values among plants globally. Again, here looking at the bivariate relationship between photosynthesis and leaf nitrogen of observations of coffee leaf traits compared to species globally. We've also explored this functional trait expression in coffee and cocoa agroforestry systems below ground. What this graph is showing here is various root functional trait bivariate relationships, so specific root tip abundance, specific root length, and root nitrogen, compared to parameters such as root diameter, showing again these trait trade-offs or these bivariate relationships between conservative and acquisitive traits. And what's interesting about this data is that across multiple countries, across multiple systems and edaphic conditions, we find the patterns of hypothesized bivariate relationships between root trait trade-offs across multiple tree crops in various agroforestry systems. And this is regardless if that cocoa or coffee plant was with a nitrogen fixing shade tree or a timber species. And we also find these bivariate relationships with coffee and cocoa in monocultures and in agroforestry systems. Now, one of the foundational tenets of trait-based ecology is that ecologically relevant functional traits or suites of traits, like the ones I've just showed you, can inform hypotheses and our mechanistic understanding of both plant response to environmental stimuli, so these response traits, as well as how plants influence ecosystem function or effect traits. And this is really where we took the research after describing the extent of variation of coffee and cocoa traits in agroforestry systems. So what this graph is showing here is multivariate analysis of these leaf traits. So pooling these leaf traits and describing them on that universal axis of acquisition to conservation and plotting where an individual plant falls on that axis to yield. And what we found, of course, is that these multivariate trait analyses explain yield production. And this gives us some clues into the physiological underpinnings of linking functional traits to yield. We've also looked below ground. This is coffee root traits. And these coffee root traits found in systems across Central America strongly explained the community structure of microbial communities and populations. So we're seeing a strong link between root trait expression in coffee and the resultant microbial communities. And finally, in cocoa, we've pooled leaf traits, root traits, and edaphic conditions along climatic gradients and find very significant causal pathways between leaf trait expression, root trait expression, and soil conditions in cocoa agroforestry systems. So there's really strong evidence suggesting that functional traits can be used to hypothesize and detect trade-offs, constraints, and synergies among crop yield and other aspects of crop functional biology. So this has led us to ask new questions that really focuses on links between functional traits and plant selection. And here we ask, in this paper that I'm presenting today, What is the role that functional traits can play in detecting cocoa clonal adaptability to local conditions in agroforestry systems? And our research objectives were to aim to provide evidence that functional traits offer new insights into the genotype by environment interactions and cocoa clonal selection in agroforestry systems, as well as we provide support for the application of a functional trait-based research to plant breeding paradigms in cocoa agroforestry. Now, to do this, working with colleagues at Cacao and CRAD, we used bivariate, multivariate, and functional trait space analyses to quantify the intraspecific variation of 9 leaf functional traits among 6 disease-resistant cocoa clones growing in 2 clonal gardens with very distinct environmental conditions. One clonal garden we classified as a mild dry season, so a mild site, and the other clonal garden was in a harsh dry season, or the harsh site. We used Cati-bred cocoa clonal varieties that were both regionally adapted to environmental conditions and resistant to frosty and black pod. Specifically, these clones were developed in early 1990s and released for cultivation in 2007. We also included the ICS 95, which is a hybrid developed in Trinidad and Tobago, which has a broad geographic range. So we set out to measure a suite of leaf functional traits among these 6 clones across 2 environmental conditions. Now, these first results are showing the morphological traits, the physiological traits, and the chemical traits in the 2 environments represented by blue and orange, and various responses among the 6 clones. And what we find is that among the 6 clones, environmental conditions had the strongest effects on leaf physiological and chemical traits, but very little influence on morphological traits. So we can see here in the physiological traits and the chemical traits that we have significant differences between the mild site and the harsh site consistently across all clones. However, we did find that when it comes to morphological traits, that we found very little difference between the sites, but consistent patterns among clones. If we look here at specific leaf area, it differed among clones with the same pattern at both sites. Specific leaf area increased with potential productivity and was highest for Catier R1, R4, and R6. Next, we wanted to look at multivariate analyses and leaf trait syndrome, so these trait trade-offs. And what we can see here is that the leaf traits of cocoa follow the generalized hypothesized universal plant economic spectrum that exists among species. So we can see that we have leaf acquiring traits such as water use efficiency, leaf nitrogen, photosynthesis falling on one end of the spectrum to the right. And we have traits such as leaf carbon to nitrogen, a conservative trait, falling to the other end. So we're seeing this universal axis of the plant economic spectrum being represented by the leaf trait expression in these cocoa clones. We can also see that the least productive clones, so PMCT58 and ICS 95, had the lowest within site functional trait variation. So we can see this by the colors of red and orange. So while the more productive clones in the blues showed higher within site trait plasticity, but lower trait dissimilarity between sites, thus really showing that these older, least productive clones have a less potential to express trait variation. Finally, we looked at trait space, and we used hypervolumes to show the size of trait variation. And this trait space summarizes the occupancy of ecological strategy of a plant or a different crop genotype. So looking among these hypervolumes of the 6 clones with various bivariate trait trade-offs, across all plants in the study, cocoa trait space overlapped for all clones. Different patterns could really be seen, though, between some clones. For example, PMCT58 showed the high disconnection between leaf nitrogen and all other traits, with two distinct hypervolumes at the harsh site, and this being represented by these separate hypervolumes seen here at the harsh site. So really distinct patterns. Another notable exception is that the older low-yielding cultivar ICS 95 expressed the lowest trait plasticity among clones and between sites. Now, this may suggest that the geographic applicability of the oldest cultivars may be limited by their lower adaptability or variation in resource acquisition strategies, despite their overall favorable disease resistance. So these cultivars may be limited in their lower variability for various strategies. But this trait space analyses within specific trait categories may be more revealing of clonal adaptability as compared to univariate analyses. And this study really for the first time shows that evaluating this trait variation may be a viable tool for assessing clonal adaptability to local conditions such as drought. So in summary, we found that clones differed primarily in their physiological functional traits at one site, likely due to water limitation, while clones differed in their chemical traits at another site, likely due to nutrient limitation. That clonal trait variation can be linked with productivity potential of an individual cultivar. For instance, the least productive clones, PMC T58 and ICS 95, presented the lowest within site trait variation, yet the highest trait changes between sites. So what does this all mean? Well, these significant genotype by environment interactive effects on cocoa functional trait expression really provide a new opportunity for selecting clones, while also providing us insights into the consequence of the selection on agroecosystem function. So what does this mean for aspects beyond yield? It also provides evidence that supports the application of a functional trait-based approach to agroforestry systems and plant breeding paradigms. So cumulatively, I hope I've shown here that these studies illustrate the role of functional traits as indicators of the effects of artificial selection on functioning and long-term plant soil feedbacks in these diversified systems. So with that, I'll thank you for listening. Of course, I'd like to acknowledge our funders, the Canada Research Chairs Program, NSERC, and all of our collaborators at CATIE. Thank you very much.